So now we're recording, I believe. And here we go. I think we're recording. Let me see who. Oh yeah, there we go. Okay. So larval rearing. Now, a couple things about how many people went to high school and took biology, and maybe even college, and maybe invertebrate zoology, and how many of you remembered all the different invertebrates and all the larval cycles and all the blah blah blahs? Because it gets pretty complicated. It really does. And I have to say that bivalves, which include well, first of all, mollusks, the phylum Mollusca, which is it's the second largest phylum on the planet. Arthropoda, the
What?
insects. What's that? Oh, uh, the mollusks are a huge phylum, including all of the snails. The gastropods are the snails. The cephalopods are the squid and the cuttlefish and the octopus. And
Maybe
then
I can get my AirPods to work.
AirPod. Oh, and mute if you're don't let the mute button get shy with you. Uh, and then there's the class bivalve. And now the bivalves are the ones that we are interested in because they include the clam, the oyster, the scallop, the mussel, and they have all a very similar larval uh, life cycle, and it's pretty complex. So this screen, this slide here shows basically uh, the larval cycle. And I want you to see a couple things about this. So you have a male and a female, or in the case of a scallop, by the way, you would have the scallop being both male and female. But in this case, it's showing what looks to be mussels, um, maybe even our bank mussels. So you can see the byssal thread there. Male and female, they spawn, they give off egg and sperm in the water column, and they're floating around. They meet each other in the water column and fertilize and will form a larvae. Now, that's not true of all bivalves. The European flat oyster, the what some people would call the bellon, if it ever lived in Bellon, that actually has a yolk sac, and the larvae are brooded within the female, which is different, quite different. Uh, they have what's called I hope you're all taking notes because this is all on the final exam. They're called leucotrophic larvae, which means like leucotho being like a yolk. But in our case, with clam, scallop, mussel, oyster, they all extrude their gametes into the water column, and a lot of them, because let's face it, what's the chance of them even meeting in the water column and fertilizing? So they fertilize in the water column, and you're going to then have a first larval stage within 6 hours. And that's called the trochophore. And I got a picture queued up of a trochophore. They're quite small. They're apparently non-feeding, so they don't need to eat yet. They're what is called positively phototactic. So that means they go towards the light. So I think of they're very fast moving. So I think of the trochophore as being a dispersal phase, because I don't know if you're up on your Greek mythology, but from Greek mythology, if you hang out with in Greek mythology, if you hang out with your parental stock, eventually they're going to eat you. And that could very well be true with mollusks, because clams and oysters are just huge filters. They're filtering and filtering and filtering. So if they spawn and the gametes go off, after a while, they're going to start filtering in. And if you're hanging out, you can be re-ingested and you won't make it very far. So, you know, to get away and get into the water column and really to start moving around is beneficial. So that's a trochophore. And then within a very short time in the life cycle, and we're going to talk about each of the species a little bit in the time, you go to what's called a straight-hinged or a D-hinged veliger. And we'll talk about a veliger in a minute, an umbonate veliger, a petaveliger. And this is as far as this lecture is going to go, is this upper part. And then, because then it undergoes metamorphosis and becomes spat. Now, keep in mind, as you look at this slide, the whole larval stage is planktonic. It's free swimming in the water column, okay? It has the ability, plankton, by the way, is from the Latin for the wanderer. And I'm not going to sing the song. I don't know all the words anyway, but the wanderer. I used to at lectures, and then people stopped coming. So I stopped doing the wanderer theme song. Okay. So they don't have control of where they're going to go. They're not saying they were spawned in Southold and they say, well, the real estate's pretty good in Orient. I think I'll go there. It doesn't work that way. They go with the tides, they go with the winds, they're all over the place. And on that note, if your oysters spawn in your creek, are you going to get oysters from there at your dock? Maybe. The larvae just kind of get caught in a creek and never get out of the creek. Oh, hold on, let me turn my phone off. If they get caught in the creek, they might go the whole life cycle in your creek, but chances are the tide's taking them out, they're taking them in, they're doing these things. So they're planktonic. And you'll see if you show up at the next lecture, we're going to talk about metamorphosis and this part, which is benthic. Because once these things finish their life cycle and metamorphose, and by the way, become spat, they're no longer free swimming. Scallops, you can say they're kind of can swim, but they're pretty much benthic, bottom dwelling. Okay, so we got a planktonic stage, and that's the life cycle, the larval cycle, and then metamorphosis. So we're going to go, last month's lecture was about the spawning. And so now we're going to do larval cycle up to the petaveliger. So again, the trochophore looks like, I have my son did when he was 8 years old, he did a video for Zoom. It was a kid's show. Zoom, Zoom, Zoom. I won't do that theme song either. But he said, and the little larvae, they look just like little hairy walnuts, which they kind of do. They look like little hairy walnuts, and they've got this apical tuft.
Who's scoring?
What's that? Oh, careful if you're talking to your mate there and you're not on mute. We're going to hear everything you say, okay? They've got an apical tuft, which I believe acts as a kind of a rudimentary rudder, even though when you watch these things under a microscope, they just spiral. They're like Sputnik. They're spiraling out of control. But they are highly ciliated. They got these little hairy walnut things, and they're pretty simple. There's not much to them. They don't even need to eat yet. Now, a trochophore only lasts a bunch of hours before it goes into its second larval stage. So the trochophore becomes a trochophore within about 6 hours of fertilization. And usually within 24 hours, it's already on the second larval stage.
I don't understand a word when they're referencing Latin.
Oh, you're not on mute, but I hear you talking about how I'm not dumbing this down enough for you yet. Yes, I'm going to speak Latin. As a matter of fact, for the rest of this lecture, I'm going to speak Latin. Okay, let me see. What was that? No, I'm not. Okay, so again.
All right, so this is crazy. And he's like, all right, so listen, I just want you guys to know, for the rest of this lecture, I'm going to speak Latin. So don't come.
Not on mute.
Dead serious.
Okay, he's serious. Here's a kind of a fuzzy picture of a trochophore, only because it's very hard to film them. They go so fast. And then again, within a very short time, they start to round out. They always, I'll tell you, no matter what the species is, the larvae always kind of look like little clams. It can be an oyster larvae, it looks like a little clam. It's a clam larvae, it already looks like a clam. If it's a scallop larvae, it looks like a clam. Oh well. What's really cool about this is you can see the shell growth every day. This line, that line right there is 1 day shell growth, right there. You're putting on shell every day. And what these are now is what's called a veliger. And the reason why it's called a veliger is it's got this organ here called the velum. And it's a kind of a combo organ. It allows it to move and to feed. It's like a little Hoover vacuum cleaner attachment here. And it's very highly ciliated. And it can go, when you looked at this picture, the velum's there, but it's inside the shell. It's retracted, probably because I picked up the larvae with a pipette and put it on a glass plate, and they're like, whoa, I'm not coming out. And so they all have that organ there. It's just inside, okay? And when it's outside and moving or feeding, that's what it looks like. So again, here you can see shell growth, the 1 day, 2 day, going out to 3 day. Here's the gut. There's some, it's highly ciliated. And these things are just capturing unicellular microalgae. It's got to be really tiny for them to eat it. What's amazing is these things now, let's say this was a 5-day-old oyster larvae, and it's an umbonate veliger, and it's got the velum and it's feeding. This thing is already eating 3,000 algae cells a day. Just 3,000 a day. By the time it reaches the end of its larval cycle, it's eating 40,000 cells a day. And that's only 10 days later. So even, you always hear that shellfish are great filterers, right? An adult oyster can filter 80 gallons a day or whatever he's reading the literature. What's amazing is, and a lot of people never get to see this unless you come into the hatchery and look under the microscope, they're filtering from day one, kind of day 1 or 2. If they haven't gotten to be a veliger, they're not feeding yet, but at day 1 or 2, they're already filtering. They're not filtering 80 gallons a day, that's for sure, but they're doing this all the time. This is what they do. That's how they get, and again, here, the velum is inside of this. You can see through with a microscope, it's still, with the light shining through, it's still pretty translucent. This is the algae that they've eaten, that's the gut. And again, here's the shell growth in a resting stage. I just love the pictures because they're so glass-like and the light refracts on them a bit. And then now, depending on the species, they'll reach the final stage, the petaveliger stage, at different times. So a clam and a scallop take about 7 to 10 days to go from a trochophore to a petaveliger. An oyster takes about 14 days. That's in the laboratory. In nature, add a week to all of those. And the reason why is because we elevate the temperature and we give them really good food. So we can actually, even at a larval stage, we can take some of the time off of the amount of time it takes for these things to finish their larval stage. They're very sensitive to temperature and food. So you can boost that a bit when you're doing it, okay? Now, the petaveliger, there's this is the velum, but there's also a foot that sticks out. Now, keep in mind, it's not 2 feet that walk around. It's 1 foot. It's more like a tongue. It comes out like a tongue, or you could almost think of it like a clam foot that's digging into the sediment. That's called a petaveliger. Okay, and that's the last stage. And there are some differences that we're going to talk about with the species, because keep in mind something. And I always try to do this when I'm describing this stuff to you. There's 2 ways that I look at these. What happens in nature, and what can you do in a laboratory situation? It's 2 different things. So nature is doing its thing. For instance, when I say to you that we have our first batch of SPAT, well, I don't know about you, but I woke up this morning in the Springs and there was snow on the ground. A little bit, not a lot, just a little bit, a dusting. All I'm saying is it's still cold out there, okay? In nature, oysters aren't close to spawning yet. None of this is happening in nature. In the hatchery, we've already got a batch of oysters that if it was natural timing in the hatchery, we would be into August for that batch. That's when in August we would have natural oysters the size that we have in our hatchery. Just so you get a perspective of how in a hatchery you can manipulate what happens in nature to have it very early. Okay, so nothing right now, none of these bivalves are even close to spawning yet, but in a hatchery you can manipulate it. Okay, just a kind of a diagrammatic of what a petaveliger looks like. Well, and all I'm going to say to you is that there aren't that many parts. There are some. I mean, this thing is still pretty small. Right now, this size in an oyster would be 1/4 of a millimeter, which is, just to give you an idea of size. So I would say when this thing is ready to undergo metamorphosis, it's at about 250 microns. Well, what does that mean? Well, a micron is a thousandth of a millimeter, and a millimeter is your window screen mesh, not your no-see-um mesh on your tent in Maine. The window screen mesh on your house, that's 1 millimeter mesh. That's the size. So these are really quite small, but even at their size, they still have all of these organs.
Seems like somebody I'd like to get to know.
Well, he just moved to Southhold.
You're not on
You
mute.
heard that part?
In the beginning, yeah.
There's a
Plays in
participant.
the process.
You know what?
Where he lives now, I have no idea. It could also be Salvo. I don't know.
I'm gonna mute you. I'm gonna mute you all.
So you're gonna stay together?
Is it Tony? No. Okay. I can mute. I might not have muted the right person.
Sorry.
Didn't that mean that I can mute anybody I want? I can just,
Yes, you can.
and you can unmute yourself, I believe. So, oh, yeah. And that's, no, I want to talk about this. Is that where I was? Yeah. Okay. See, you guys threw me all off here. How do I get rid of this thing? Oh, there it is. Okay. Here we go. I'm getting, here we go. Let's go back here. That was that. This is this. I'm trying to move this thing out of my way so I can see. Oh, there it is. Chat. Now, this is kind of an interesting thing that I want to bring up. I was saying to you that when I say, by the way, when I say in these lectures or whatever they are, love fests, a clam, an oyster, a scallop, or a mussel, I'm talking about ours. So the eastern oyster, that's the oyster we grow. The northern quahog is the hard shell clam that we grow. The Argopectinuradians is the bay scallop that we grow. So there are lots of different clams and lots of different scallops and lots of different oysters. I'm talking about ours. So when I say that all of these things have the same larval cycle, that's true. Here's a really interesting little tidbit for you. You've probably heard of the zebra mussel. The zebra mussel came into the Great Lakes from the Baltics, and it really exploded in the Great Lakes, and it just mute you. How did the zebra mussel get to? The zebra mussel is a freshwater mussel. Okay, Great Lakes, freshwater. Zebra mussel, freshwater. Now, the larval cycle of a lot of freshwater bivalves is very different to marine bivalves. Let's take a blue mussel, or the one that you like to eat, or the bank mussel that you see along the creeks. Those have the larval cycle we were just talking about. A freshwater mussel and a freshwater clam has a totally different life cycle. And what part of the life cycle of a freshwater bivalve is called a Glochidia larvae. And a Glochidia larvae sticks in the gills of fish to incubate. Okay? Now, what the heck's going on with a zebra mussel that came across the ocean in a Baltic trader in the bilge water? Because that's how they got there. The Baltic trader goes up, sucks up all the water in the lake there for ballast, steams across to the Great Lakes for grain, dumps their bilge water into the Great Lakes to get rid of the ballast, takes on grain, and goes home. And when they blew that bilge water into the Great Lakes, they dumped, apparently, an entire vessel's worth of zebra mussel larvae. Now, if zebra mussels had the same larval cycle as the freshwater mussels in the Great Lakes, that bilge water would have had to have a whole bunch of fish with the larvae stuck in their gills. But it turns out that zebra mussels, which are a freshwater mussel, have the exact same larval cycle as our marine mussel. And that's why we got blasted by them in the Great Lakes, because nobody realized that the bilge water was like a big larval rearing vessel. And it dumped these larvae that were, they must have said when they got there, "Whoa, I don't know where we are, but this is awesome." And they went, bing! And it just exploded with zebra mussels. So much so that in the Great Lakes, they have power plants, and they have a different system than we have. We're used to things like this, and we would call mussels, by the way, a fouling organism. In the Great Lakes, they have the intake lines for the power plants were 12-inch pipes, okay? And within a few months, they became like half-inch pipes. The rest of the opening was all zebra mussels, and it shut down the power plants because it was a fouling organism, and the power plant flow was so good. It's exactly the flow that these shellfish wanted for feeding. So it was a real environmental monkey wrench in the Great Lakes. It shut down the power plants. What it also did is these bivalves are tremendous filterers, and they scrub the water clean. All the submerged aquatic vegetation. You remember back in the day in I'm about the same age as everyone else, and you remember hearing like you could light the lake on fire with a match, and this, well, those days are actually kind of gone because there was a lot of filtering going on. What was kind of funny about that is you'd see mountains of zebra mussel shells on the beach washed up. And they would say, "Hi, Tony." They would say, "Well, why don't we just take those zebra mussels and use them for compost?" Well, the first year or so, they were more like toxic waste because they had taken everything out of the water and lay up on the shoreline. So just an interesting tidbit that these freshwater mussels had the same larval cycle. Now, once we have our larvae, we have these larval rearing vessels. We call them conicals. And so the wall of the conical is straight, and then here's the break in it, and this is like a cone that goes down to a little tiny 3/4 inch opening at the bottom. And we filter our water. This is a filter bag, and we put the water in. If anyone wants to get involved in the hatchery, rearing of larvae, we do this kind of early, Monday, Wednesday, Fridays, and we'll be doing larval rearing for the next couple months. So you can get involved in it if you would like, because it is a little bit of a routine. We have to drain these tanks every Monday, Wednesday, Friday, refill them with heated, filtered seawater, and they need to be fed every day. So we raise them in here. These are 100-gallon. And so here's somebody, oh, we got the little rubber ball as the spy. If you hear the rubber ball bouncing on the ground, that means you overfilled the tank and the ball fell out, which happens like all the time because SPAT members love to chat and not watch the rubber ball coming up over the horizon, which says, warning, warning, you're about to overfill the tank. Now, keep in mind, the larvae aren't in the tank yet. That would be really bad. If you put the larvae in and then filled it up, then they're going all over the place. So we fill these tanks up, and we have to keep the water. Then we add the larvae back in. And how do you get the larvae? I mean, what we do is we drain the tanks out. Oh, what does that say? Yes, beware of aggressive larvae. That's actually, I recognize those feet. That's Charlie Peck. What's he doing in the tank? This is my mistake. That was me in my hoodie day. That was back when I was a rapper. Okay, there's Otto. We've been doing this a while. What he's doing actually was my bad. I was fixing the bottom of the conical with epoxy, and I epoxied the cork in, and he's trying to get the cork out. That's what that picture is. Every Monday, Wednesday, Friday, we drain the tanks down into screens. There's a piece of sewer pipe with a screen on the bottom, and that what looks like paint stain, that's 1 million larvae. And you can see the color is the color of the algae that they've been eating. These are so small, they're not even the head of the pin, they're still the pointy part of the pin. But you can collect them in the screen. We have every screen size. You can collect the eggs, you can collect, and by doing this, we're cleaning the tanks every Monday, Wednesday, Friday, but we're also collecting all the larvae, and now we can size them, we can count them. They are totally countable, so it's not guesswork. And so that's what we're doing here. When I say it's not guesswork, it's because you have to feed these things every day. And so you have to know, and their demand for algae changes every day as they're growing. I said at the beginning, they start by eating 1,400 cells a day per oyster larvae, 1,400 algae cells a day. And by the time they're a petaveliger, they're eating 40,000 a day each. So you have to adjust the food that you're going to feed them. It starts off with 1 liter at this density, goes up to a 10-liter bucket at the end. Or you could do it the way I do it, which is my own scientific term of eyeballometrics. Just throw it in there, No, it's actually not the way to do it because you can overfeed these things. If you think about brown tide, and nobody likes to think about brown tide, but we had brown tide. And why did the larvae suffer? Why did the shellfish suffer so much in brown tide? Was it because it was toxic? Or was it because it bloomed? It was very small and it bloomed in huge, huge densities. And there's a possibility that it became, have you ever done this? I just did this the other day by mistake up in Cape Cod. I would never do it at home. There's Kimmy in the background. She'll attest to this. Have you ever taken your Electrolux vacuum cleaner and vacuumed out the ashes from your wood stove? I did that the other day up in Cape Cod, and it shut the vacuum cleaner down immediately. And the little warning sign came on saying, no, way too dense of really fine dust. You lose. Okay, that's what happens with shellfish too. If you overfeed shellfish, they're filtering and they're filtering and they're filtering. It doesn't mean that they're like, oh, this is great. This is like being at the all-you-can-eat buffet at the Olive Garden, which could be fine for some people and devastating for others. Okay, so you don't want to overfeed and you don't want to underfeed. So it's not really guesswork. You want to make sure that you're kind of being appropriate with this, with the feeding regime too. Because you must feed them every day, okay? We're like little zookeepers when we're raising all this shellfish until, now keep in mind, and I failed to mention this at the very beginning, but, and I mentioned this probably at the first lecture about algae. In aquaculture, you have 3 stages of aquaculture. You have the hatchery, you have the nursery, and you have the grow out. The hatchery phase always requires cultured microalgae. You have to grow the food for the hatchery stock. That's what they eat, okay? The nursery is the time where you can stop having to feed them and they go on ambient water, in effect. That's a true nursery. And that's coming up, hopefully, knock on wood. Can you hear that wood? Hopefully, let's say April 15th. That's the water temperature came up enough, algae starting to grow, these things that are eating us out of house and home because we have to grow, it's literally have to grow this and we're feeding them and feeding them. We can now put them on ambient flowing seawater and nature will feed them and they go on auto feed. That's the nursery. But we're in serious hatchery phase right now. We need to grow a lot of algae to feed all these little critters until the water's warm enough and the algae's up for them to grow. That's just living in the Northeast. That's the facts, okay? If you put them out too cold, they might not die. Oysters, clams will probably die, scallops would die. Oysters might be able to go into a kind of a hibernation phase, but they certainly won't grow and they might die. So it's not like I can put these on ambient flowing seawater anytime soon, even though April 15th isn't that far away. Might be May, we'll see. And again, once you have these things collected, you can size them. They, like your oysters that you're growing in your garden, even larvae, they don't grow uniformly. Some are larger than others the whole time. And so you can kind of gauge that because there you're looking at a bucket and all this stuff on the bottom are oyster larvae and some are swimming around and some are on the bottom because this is late stage larvae. And that bucket is 1 million. Each conical that I said was 100 gallons can grow 1 million larvae in the conical. That's what we try to set it at, 1 million larvae per conical. We have 8 conicals in the SPAT shack, so we can theoretically grow a batch of 8 million larvae to go into setting. And you might say, yeah, right. Like you really know how many are in the bucket. Okay. And if you say that to me, I'm going to make you come over to my place and count them. There it is. Because that's what Ed is doing here. What you do is you can count larvae. You can count larvae. Okay. And don't make me mad because I can really screw up your eyesight. Okay. And I'll show you why. Let's say that you want to count all of the larvae in this bucket. How would you do it? Okay, Here's how you would do it. You can see here, this line here, that's 5 liters. This line is 10 liters. We mark them because, I mean, my eyesight's really going shot. I can't see anything anymore. Turns out I have 20/20 vision, but I need 125 readers. So I can't see anything itty bitty up close. I can see the sign that says you're definitely speeding. Okay, but so what you do is you take this bucket of salt water. Obviously, this is always salt water. And you fill the bucket up to 10 liters. Okay? Then you toss it back a bunch of times. And what that's doing is assuming that you're having a homogeneously, what's the word I'm looking for? What is the word? You toss it a couple times so that when you take a sample, you're going to get a uniform amount in the whole bucket. So they're evenly dispersed, a homogeneous dispersion of larvae in the bucket. This little graduated cylinder there is really just used as a plunger to really make sure that the whole 10 liters with the larvae are well agitated, so that now when you take a pipette, 1 milliliter pipette, you draw 1 milliliter up, you put it in a special, I don't know if I have a picture of the, so he's agitating it. Oh, and you can't really see it. And this is, I love this photograph because this is a picture of Otto in 2002 in the original SPAT Shack. And look at that microscope. I don't even think it has a light source. That's a classic photo. But on the bottom of this, I guess I don't have a picture of a Sedgwick rafter counting chamber. It's a little square slide with a border around it and a grid in it. And you pull the 1 milliliter sample, put it on the Sedgwick rafter, and then you can use a microscope with a mechanical stage and a tallywhacker. Click, 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 so you don't have to keep track. You see a larvae and you click, 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 and you're going across the screen and you count the entire milliliter. And let's say you get 125 larvae in your count. How many larvae are in the bucket?
125,000.
You know, for a doctor, Robert, your math sucks. Try again. 1 milliliter from a 10 liter bucket, you got 125.
1,250,000.
Thank you. But don't get me wrong. My math is the worst math of anybody's math. Okay. I only know that because I've counted so many freaking Sedgwick rafters of larvae. And now here's why you never make me mad at you, because early on when you're doing these spawns, and I've done this a couple times, I actually just did it to Otto again this year. You take a sample out of a bucket from day 2 of your spawn, and you don't really know how many larvae you have yet. You can pull a sample out, you put it, the 1 milliliter under the microscope, and you leave the member to count them. And you go to have a cup of coffee, and you come back a half an hour later, and they're still counting it. They're up to 2,349.
Okay.
Okay, so now you have 2,000. Now you have, like, you know, a 500 million larvae in the bucket, and that can happen. So, I mean, you wouldn't do that. You would immediately look under the microscope and you would see these swarms of larvae and you would say, it's uncountable. Not, oh my God, he left me here to count all these larvae. And I never kill my larvae. You're supposed to put a little drop of iodine in and kills them all so they stop moving around and you count them. What you would have to do in that case is take the 10-liter bucket, agitate it a whole bunch of times, maybe pour off 1 liter of it, fill it back up to 10.
What am I drinking?
And count it again. You'd have to do a serial dilution until it's countable and keep track of it. So, but if you
Tetrault is really on tonight.
got 125 larvae in the
His wife, I got
sample,
that.
you had a
I
million.
was just talking about, he was just 8 times.
You hear everything you're saying? We hear everything you're saying, and you're going to get in trouble because you're probably going to say something that's not appropriate. Like me. No, I have to watch myself all the time. And you can, we have a television. We have this is all very old slides. I love these old slides. Now the SPAT Shack is all insulated and covers, and we got a new microscope and a new camera. But you can look under the TV if your eyes are bad. And we write everything down. Oh, I'm going to mute you. Where are you?
The hatchery, feeding the larvae,
Ethan.
getting the larvae.
Oh, it's not Ethan. Oh, it was. I muted him. I love this thing. I wish I could do this to everybody I wanted to mute. Somebody called me.
You can mute the whole class. You
Yeah.
can mute everyone at once.
Oh,
You
As
can.
the host.
Hey there.
I should give you control of the mute everyone at once button. I'm still learning. Darcy's been busy. We don't get to bond much anymore. She could teach me all this stuff, so I don't. But I did learn from last month about not having the little weird microphone speaker that was screwing me up. You can hear it better now, right? Okay. So we document everything so we know how they're growing and what they're eating. And by the way, you also have to adjust their feed as they're growing bigger. They'll start off eating very small, what we call T-iso or pavlova, 2-micron algae that they can eat. And later you can add a diatom, and later on you can add a chlorophyte. So you can mix their diet up as they're growing. But you have to really know what it is, because algae are not equal. Back in the day when they were trying to figure out this new really high test algae, they were killing all the clam larvae because they couldn't eat it. You remember the expression, never eat anything bigger than your head? Well, these guys didn't really have that down yet, because they were trying to feed a 20-micron algae to a 70-micron animal. And it's just hard to digest, as they say. One day we're going to fool around. We wrote a grant to look at continuous flow, where you might say to yourself, how come you grow 1 million larvae in 100 gallons? Why don't you grow, 8 million in 100 gallons? And I've tried it. I've tried everything at least once. And they won't grow. It's the weirdest thing. I'd love to know exactly why larvae have an issue when you put too many in confinement. It could be 3 different issues. It could be that there's a toxic buildup of the metabolites. That's a possibility. It could be not enough food, but you can add more food. So I don't think that that's the issue. But what I think is the issue, this is my half-baked theory. I've got a lot of half-baked theories, and this is one of them. These things are swimming around like atoms. They're going all over the place. If you put a lot of larvae in a kind of a confined volume of water, there's a good chance that they keep bumping into each other. And every time they bump into each other, they're going to stop feeding. So if they're continuously bumping into each other, it might really be interrupting their feeding. So you wouldn't think that something that you can hardly even see with the naked eye would ever be space limited. But, so there are some experimentation we can do with, this one wouldn't address it. The volume of water is the same. What this one addresses is continuously adding bits of new seawater and new algae. So this is a screen that keeps the larvae from flowing out, but the continuous flow of water and algae can go out. So we've messed around with that. I haven't had much luck with it yet, but I'm spurring people like Chris on to want to experiment, because every time I see him, I can tell he wants to do a crazy experiment. And late stage, now here's the thing. Oh, and I guess we haven't gotten into this yet, but I'm hoping we do. How do you know when they're about to undergo metamorphosis? And why do you even care? one reason you care with oysters, and we're going to talk about this a lot next month when we talk about post-set, especially oysters. Oysters, when they undergo metamorphosis, they cement to structure. That's their modus operandi, okay? That's bad Latin. I don't know. I said it wrong. I know there's a Latin guy in the crowd who's going to say, that's not how you say that. Anyway, if you're not paying attention to your larvae and they set all over your conical, you're going to be in there like that picture way back with the feet sticking out with a Scotch-Brite, trying to get them off. Hey, Bill. Trying to get them off. Oh, Bill's laughing.
About daylight savings, Kim. I'm sorry. I was waiting all day long for this meeting, and
You know what's funny? I even told the person from the United Kingdom that it was daylight saving.
That's what you.
Oh well. I guess
Man, I waited all day long for this.
No worries, because Bill actually just came on board, but he's done more larval rearing as a SPAT member than just about anybody. He's spent a lot of time in the hatchery. But the thing is, if this larvae is about to undergo metamorphosis and you're not paying attention and it sets all over your gear, you're going to be Scotch-Briting it off to try to save it. And it's a pain, whereas we're going to show you techniques of how to actually corral them and capture them so that you can control the metamorphic process. So this is what that contraction is. It's what's called a downweller within the conical. So that we're going to get into detail about that next month. But and again, you can clean these things. But oh, you know what? I really didn't talk about this at all. So I wonder where I should go to talk about it. Oh, I know I didn't talk about it because it's next month. Okay. I was going to say, well, you didn't talk about how do you get ready for these things to undergo metamorphosis? Because, and it's next month's lecture. It's the whole lecture. So I shouldn't give you too many things, but it's going to be based on the size of the larvae and a whole bunch of diagnostics that gives you a very clear warning that you have to pay attention that these things are about to undergo metamorphosis. And so what you're going to see next month is the larvae searching around for structure to cement to. This is a shell chip, what we call microculture. And I don't know if you saw the video that Darcy posted, but I think I have. Let me, I'm going to stop sharing. I'm going to go to hold on. I'm going to go to my.
Oh, you're in your house now.
Oh, yeah. Well, I'm in a house.
That's funny.
I don't think this one runs. Does it run? Where am I? Hold on.
I thought you were at the hatchery.
Let me close this. I'm going to queue up a movie here and share it again. Hold on. And share screen. Where's my share screen button?
Oh, wow.
Sorry, hold on. I'm almost there. A share screen. Here we go. And so this is a post-set oyster. The shell, you can see it moving, the gill structure. Let me hit and now you're going to see, here's the larvae swimming around at that size. There's a pediveliger swimming around. What's interesting about that is these are the same age. Just get that visual in your mind. This is the new shell growth. You can see the soft tissue moving. This is the gill structure. You can see it all moving.
Oh, yeah.
And then here comes the larvae swimming around. That's the same age. It just hasn't undergone metamorphosis yet. Here's one that, let me show you that one more time. The amazing thing about that is here's the size of the larvae right here. You can see it. Right here is the outline of what looks like a clam. And all of this is brand new explosion of growth. And that's what we're going to concentrate on next month. And it's really, even after doing this for over 30 years, it just blows me away in so many respects. It's so fascinating to see that, and something that you can do with us too. You can do this whole process with us, from spawning to raising the larvae to setting them and looking under a microscope. It keeps me going in the profession, and it's kind of kept SPAT members in the program, because it's really fascinating to take 30 oysters and make a couple million out of it by these techniques, and actually get to witness it, because you can't see this in nature. I mean, you just can't, you can't see this process happening in nature. It's impossible, even with 125 reading glasses. It ain't gonna happen. You can only see it in a kind of a laboratory setting. You can do a plankton tow and look under the microscope of wild larvae. They do that all the time to see what kind of critters are in the water column at any kind of given time. But it's really quite fascinating that you can do that. And it's really, to me, also fascinating that you can actually do this without, a crazy amount of expense or even training. This is something that we teach SPAT members to do, and every year do this. We create our own new SPAT. And so, I encourage folks to get involved, but you don't have to. A lot of newcomers to the program see these lectures and they're like, but how are we going to be growing our larvae to get our oysters? I mean, our kitchen isn't big enough. No, you don't have to, you can come and just get your seed. You don't have to learn. There might be a final exam, but you can totally fail it and you're still going to, be a member. You don't get graded. Well, maybe you do. I'll grade some of you just out of spite. I'm going to grade Chris, because he should know that. But that's basically the larval rearing process. And if nothing else, it will bring you back to, let me stop sharing. There we all are. If nothing else, it'll bring you back to your high school days of when you were supposed to be remembering all these larval cycles. And now you only remember them when they're on some bizarre crossword puzzle. I've never seen trochophore as a word on a crossword puzzle. I don't think I've ever seen veliger on a crossword puzzle. Has anyone ever? But you have seen, but I take that back. Just last week, pupa, which is a larval cycle of an insect. So there you go. So there's some larval cycles you're supposed to remember. It's all good stuff. And, again, there aren't that many, oh, Tony's, who's that? Samantha? Lack of oxygen? Possibly. That was a question about why you could only grow so many animals in a conical. What is the limiting factor? Here's what I'm going to say as a sales pitch. I don't think there's another facility that I know of where community members or just anybody can come to ask questions and actually do some experimentation at basically no cost. I mean, any questions that you have on this stuff, we can develop projects to look at them, including changing the stocking densities of larvae, or how can we improve on metamorphosis? Are there structures that they like to set on better than others? That's one I really want to work on, because, and what I guess what I'm saying is most other places are so confined through either funding or time or all these things that a lot of things can't get done. And we're not in any rush. I've been doing SPAT for 21 years now, and I figure I've got at least 9 or 10 more years left in me. So if you want to do these projects, you better step on it because you might only have 9 or 10 years left to put up with me to do it. And then, then we'll see where it goes. Somebody's going to inherit the program. It's been a good program and we want it to keep.
You might have 20, Kim. You might have 20 years left.
If I'm doing SPAT at 80, it only means that I'm doing it with all the other 80-year-olds that are in the project. So you could be right, Bill. I don't know yet. You never can tell because it's a lot of fun. It is a lot of fun. And I highly encourage, this whole COVID thing threw a little bit of a monkey wrench into the program, but we still operated. And I think that, I follow the science too. But, it sure looks like come 4th of July, we're going to be in a lot better shape for people to be. We still are masking up and let's face it, SPAT is outside and we're doing, there can be 20 people at SPAT, but they're all separated into little places. So, I feel pretty safe being with everybody. It's been a challenge but the vaccines are around and I encourage people to do that. And we're going to be getting back to normal SPAT. The trouble with normal SPAT is utter chaos. So, which is fine. I mean, it is, it's fine. It's a lot of fun. It's a lot of fun to have people come out and we have lots of activities going on now. We've done a lot of building. We rebuilt the docks. We're going to start hopefully rebuilding a lot of the pilings so that we can have a more secure garden space. Tiana is signing people up now. We've opened it up to 60 people this year. So there's a lot of activity going on and I look forward to seeing everybody. Hopefully we can do, who knows, maybe we can even do the SPAT party this year. I'm hoping, and celebration come winter. So if there's no question, if there's questions, now's a good time to unmute and ask the questions on any of that stuff.
No, Kimmy, they just said they're going to do the celebration.
Well, they said that last year because we did a virtual celebration. So I'm not sure they're going to do a live one. They're already saying a live one.
Yeah, they're saying a live one. Yeah.
Well, that's awesome. And that makes sense because, by December, we really should be in.
Yeah, we should be moved. Yeah.
Anybody? Anything? Otherwise, it's dinner time up for everyone. Is this a good time for people for these things?
Sure. Yeah, definitely. I've never been on Zoom. I mean, I'm not a Zoom fan, but
Your cat is though. I can see the cat zooming away. Yeah.
Can you see him in here? The kitty
Yeah,
cat.
yeah. He's right in your left ear, right ear. Good. Okay.
No question. What happened to the scallops last year?
Oh, a question about scallops. Okay.
I know we were off, but did they have a die-off? Were they okay or?
Oh no, they had a die-off. Just so you know, this is 2 years in a row of a pretty significant die-off of the adults before harvest. Okay, not all the scallops. That's an important thing to note. What's happened for 2 years in a row is the scallops have spawned, the new stock has been created and lived, and the adults died before harvest, 2 years in a row. Now, so it's not the same disaster as brown tide because they spawned and the next year's stock is there. The dilemma is, here's an 18-month creature that you can harvest every one of and it won't screw up the fishery because they're going to die. And you don't get to harvest and eat them because they die just before opening day. So because of that, with Cornell and partners of Cornell, there's a lot of research being done on what's causing this. Personally, well, I will preface this with something I said before. I'm full of a lot of half-baked theories. So this is my half-baked theory. that the water temperature has gotten higher in the summer.
It's temperature.
And the temperature right after scallop spawn has been inordinately high. And I think that's stressing out the adults, and that's what's causing it. But there are two other potential issues that they're looking into. One of them is it's either a bacteria or a fungal or a viral issue that they're looking at that could very well be playing a role. There's also a new predator on the block, the cow-nosed ray. Now, I find it very hard to believe that the cow-nosed ray is causing all the die-off. But when you have all three of them happening, you can get what I'm going to call a pan-bay phenomenon, which means the die-off seemed to happen throughout the Peconics. It wasn't in one creek, it wasn't in one area. So they're looking at it, and it's frustrating because I think I was even quoted in the press as saying, I'd be very surprised if it happened next year. And it happened the next year. So that kind of screwed me up. I thought we had just had a really very, very warm event right after a spawning event. So it could be a little bit more intense than that. And I don't think that it's the end of scallops, but it's frustrating as all get out because scallops are a cherished species and very hard to.
You know what I think, Kim? I can jump in here.
Yeah.
Because they're much more sensitive animal.
They are very sensitive animals.
And the oyster, they're like as strong as.
Yeah, well, I mean, there's no doubt that the scallop is a vastly more sensitive animal. So anyway, we'll keep you posted on that. And I think, and maybe Darcy knows this, I think we're posting this new scallop research information on our website. It's coming up.
Yeah, I have recently Steve Tetrault gave me some updates for the scallop page, and hopefully we'll have some more, and whatever I get, I put up as soon as I can.
Yeah, but I do know, I spoke to Steve this week, and I do know that there's a lot of new research on the horizon for this year with Stony Brook and Cornell and a couple other groups. So we'll keep you posted on that.
Did the die-off just happen in Peconic, or was it other parts of the country or the coast?
No, I think it's just our area. I'll double check that, but I will tell you right now, there's hardly anybody that has scallops left. So the only place that I would go looking would be Martha's Vineyard, Nantucket. And I really can't think of another place that has bay scallops anymore. Sea scallops are another issue altogether, but the bay scallop is really, really a precarious beast. You won't find them to the south anymore. They're gone from Florida. So, you know, it's not like when you say Peconic Bay was like one of the last strongholds of the bay scallop. So we'll take a peek at that, and I'll keep you updated. Good.
Kim, hi, it's Robin from the UK.
Oh, there you are.
Hi there. Going back to the larval life cycle, do you have a picture of what the cues might be that allow or prompt the various larval stages to move on to the next stage?
That's really, really interesting. As far as cues, I would say that it's not so much, when you get to the petaveliger stage where they're going to go through metamorphosis, which is next month's lecture, absolutely. And we're going to talk about that in depth. As far as the intermediate larval stages, that's really interesting. And the reason why I find it interesting is when you're culturing these things and you're looking under the microscope, not only do you see each larval stage, but you also see kind of intermediate larval stages. So for instance, the fertilized egg to the trochophore, there's a stage called the morula, which is just a gyrating group of cells. It's not even a larval stage anymore. When you see a trochophore going to a D stage, you can see this chaotic motion going on. And same with, so there might be chemical changes that are cueing it. I would say that
No, it's temperature.
no, it's not just temperature. It isn't just temperature. Temperature plays a role. No question that temperature plays a role. But I'm going to give you a for instance of that kind of alludes to something going on with a cue. When I said that we grow 1 million larvae in a conical, one year I put 8 million larvae in a conical. And after day 2, they didn't go to the next larval stage. At day 3, they didn't go to the next larval stage. At day 4, I threw them away. They just weren't going to the next larval stage. So now something was going on that was keeping them from going to the next larval stage. Was it a cue? Was it a limiting factor? Maybe it wasn't temperature, because it was at same temperature. It probably wasn't food, because we put in plenty of food. So was it a metabolite that might have been a chemical? Was it just general health of some genetic quality that the eggs weren't even healthy enough to go? It's very intriguing. So I wish I could give you a definitive answer on that, but I would say it's physiological more than it's chemical. That's my thought, is that it's really let's go back to humans. When we're growing, we don't really take a cue to grow per se, or do we? Is it a chemical that allows us to mature and to grow bone structure? And so, it's amazing also on that note that something as simple as a little larvae that you can even hardly see does use chemical cues. So I wish I could answer the question if there's a cueing that does that. Part of it just might be, I'll tell you this. Here's another intriguing part about it. You can manipulate them to move faster. We've done that using different temperatures and different diet. So for instance, there's an algae that's a very high lipid strain of algae called pavlova. And if you feed larvae this and we can do, here's a perfect experiment, is looking at that, which I've done, but I haven't done it enough to prove it definitively. But this food allowed larvae to undergo metamorphosis days in advance. So was that a chemical cue that allowed it to advance faster? Was it just a nutritive issue that allowed the tissues to grow faster? The bottom line is you can manipulate these things into slowing down or speeding up. Temperature, certainly. If you run them cold, it takes a lot longer. So I think it's more of a physiological than a chemical cue, but I'll look that up. I don't think I'll find it in the literature. I don't think anyone will be able to define that quite yet, but I love to look at questions that I don't know. I can't even lie to you properly. Okay.
Yeah, great. Thanks for trying anyway.
Well, and I will follow that through. But if you can make next month's lecture, you're going to be blown away by the cues that cue metamorphosis. I'm not going to give it away yet. I spent my whole graduate thesis on it. Took me 3 years, so I won't bore you for 3 years with it. But that is about chemical cues, and so stay tuned for that.
Okay, great. I'll be there.
You're good. Anybody else?
I have a question, and anyways, I look forward to becoming a SPAT member, but But anyways, it was really what you covered last month where I wasn't there for it, but you brought it up today that it's early in the year, and even the next month or two is early in the year for spawning for a lot of shellfish. And it reminded me of something I've been curious about for a year, where I was digging steamers or Maya last year and at Easter time, and they were clearly incredibly more ripe than I've ever tasted before. I didn't even like the taste so much, even though I like them almost as much as I like scallops. And the question is, if you're running into clams that are ripe, do they stay ripe for a month or 2 months or?
Oh, okay, so when you say Easter, so that was in April.
Right.
And depending on how that April was running, what you were finding, and I would also say that Maya, when you say that they were ripe, they were ripe, and they're hard to eat when they're that ripe. They're chock-a-block of spawn, and they're really hard to eat.
It was really rich and fat.
But the interesting thing about that is that I would venture to say that this is the case, that Maya, what you were seeing was what we call a conditioned brood stock. That's the kind of term we would use. That means that this adult had fed really well in the spring to get ready for spawning and was ripe. And I would say that Maya does that earlier than the other shellfish, that they are actually one of the species that spawns pretty early. So when you pull that thing out, that can also be a function of the water temperature of that season. If it was a particularly warm April, they were feeding probably in early March and putting on this gonadal mass, which they do with temperature change, ripening up the gonad. They put on gonadal growth during that time. That's what you were seeing. And you're pulling it out and it's ripe. And if it was really ripe, the first warm day after Easter, they would have spawned. That's their cue for spawning. The first warm day would set that thing off hair trigger. So how long does it last? It really doesn't last that long. They don't do a lot in their life, but what they do really well. And so what you were witnessing was a species that conditions early, that will spawn early on the first, and when I say a warm day, it could have been, let's say, 50 on Easter, and one day of 70, where the water temperature in a shallow creek warms up quite dramatically, that just that day will set them off to spawn. And, if they didn't, they'll keep conditioning until they spawn. That's what they'll do. And once they spawn and they're spawned out, they could recondition, but it'd take a while. But that's what you were seeing. And I've seen the same thing happen in that time of year with oysters, where you can hardly even eat them. Not April, maybe May, where you don't even want to eat them. They're so creamy looking and just, and you shouldn't really. You should let them spawn in nature. So good.
Thank you.
Good. And keep that. The best part of this whole program is your own interest that you do, going out clamming, seeing these things happen, maybe picking up a ghost shrimp or a big ribbon worm and just saying, whoa, and what's going on with this and what's good. That's what we do in life. That's what keeps us interested. And when it comes to shellfish, you should have all, except for the queuing of the larval stage, you should have all your questions answered.
Is that this?
Good?
Kim, who's in the background there? Is that your stepdaughter?
Yes. No, that's Kimmy. That's my wife.
That's Kimmy? I can't even recognize you.
Yeah, she's very young looking.
Oh, that's really nice.
Yeah.
You look so beautiful.
I'm actually watching the lecture here.
Oh.
but
Oh, okay. Oh, wow.
The audio live.
Yeah,
I get live audio.
Oh, wow.
Hi, Darcy. I see you too.
There's Darcy. I'm going to stop recording, though. No, are we done? Are we good? Everyone's going to dinner?
Yeah, go eat your dinner.
Good. Thanks, everybody, for coming out, and don't be shy. Come around and say hello.
Darcy,
Can I say one thing to Kimmy?
Yeah, no, we'll do that offline.
All right.
Offline, okay, I hear you, I hear you.
You stay on and
Thank you.
see ya.
Darcy, let
Bye,
me-
guys. Have a good weekend.
all right.
All right. Thank you. Thank you.
See
indeed.
you, Robin, we'll be in touch.
Yeah, great.